Hey everyone. These next couple slides are to give us um, a brief overview of the different organelles that we can find inside of our cells. Before we go over those, I just want to quickly discuss what an organelle is. So an organelle, the way I think of them, they are the organs of our cells, right? So in the human body, we aren't just the human, right? As we mentioned in chapter 1, there are many components to a human. We have a heart, right? That has a function. We have a liver that has a function. We have our spleen. I can keep going on and on. Those are the organs that have roles that allow us to function and keep us alive. Just like humans have organs, cells have organelles. They have their own like mini organs. All right. So with that said, we're going to go over a few of them here. The first couple on these slides are the endoplasmic reticulum. It is going to be connected to the membrane-bound sacs, canals, and vesicles, and they are a little transport system within the cell. There are two different types of endoplasmic reticulum. We have the rough ER and the smooth ER. The rough ER is going to be loaded up with ribosomes, which we'll talk about next. And they're going to be a site where protein and lipid synthesis are going to occur. Protein synthesis, you're making proteins. Lipid synthesis, you're making lipids. All right, so these are really important. Smooth ER is also going to be used for lipid synthesis, making fats, but also breaking down of various drugs, right? Different chemicals. Ribosomes are also extremely important in the body because these are free floating or connected to the endoplasmic reticulum, as you see here, the rough ER. And these are going to be the site where our proteins are going to be made. And that is very important, right? Again, so what are we made of? Proteins. Where are they made? In the ribosomes. A couple other examples. We have the Golgi Apparatus. This is a group of flattened membranous sacs, and their job is to package and modify proteins so they're ready to go. So this is not a great example, but I think of this kind of like the UPS of our cells. So we make the proteins in the ribosomes, and then we take those ribosomes, we modify them, we get them ready, we prepare them, and we put them in packages. Not literally, but we package them, we prepare them so that we can ship them to other parts of the body. Right? The mitochondria is the powerhouse of the cell. Everyone always says that, right? And it's true. This is where energy is going to be extracted from glucose. So that is really important. So that is where cellular respiration will occur. The next few are a little less common. Lysosomes are really important. These are enzyme-containing sacs within the cell, and their job is to digest and break down unwanted substances, as well as worn-out parts of the cell. So these are kind of like the recyclers of the cell. So if there's any damaged parts of the cell, we break them down so we can use them for something else. If there's any unwanted substances, we break them down so we can use them for something else. Peroxisomes, similar functions. They're also an enzyme-containing sac. The difference is they are going to more specifically break down organic molecules. So other things that have come into the cell that we can break down, usually for energy, to help us out in that regard. Centrosomes are another component, and they are super important. They are two rod-like centrioles, and their job is to produce cilia, which are like little like hair-like projections. I think of them kind of like ropes. And their job is to help out during cell division. These are going to be used to help make sure that we organize our chromosomes and pull them apart so we have equal amount of chromosomes in each new cell. So they're very important for that. We'll talk more about those when we get to the cell cycle. Lastly, we have cilia and flagellum. People always confuse these two. Cilia are short hair-like projections that are on the surface of the cells. And flagellum are also on the surface of cells, but they're more like a tail. So cilia can be used for things like making sure that there's a high surface area. We can see this, for example, on the inside of our trachea, right? So we have a whole bunch of these little hair-like projections. So in case we accidentally breathe something in, like maybe, I don't know, some sort of pathogen, maybe some dust, that will get trapped on the cilia, those hair-like projections. And if you get enough of it on there, your body will have a reflex and cough it out, right? You don't want to get that stuff down into your lungs. It's gross. And that's one of the issues. People know a lot about cigarette smoking. The tar that comes out of the cigarettes will actually cover these and make people more likely to get sick from various pathogens getting into their lungs because they're not being stopped by the cilia. Flagellum are these long tail-like projections that just go back and forth. They're like little tails. This is going to allow for motility in cells. Specifically, this is like the tail of the sperm. So those are a few different components of the cells. There's definitely more. But this is a nice little overview, and I want you guys to take some time to look over the different components of the cells. And if we have any questions, we can talk about these during our lab.